Hello students, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. I am going to talk about a module that is hydropathy plot and membrane asymmetry. This has been taken from paper Membrane Biophysics. As you know that membrane or plasma membrane is composed of phospholipid in the form of bilayer along with the protein embedded into it, and distribution of these protein in the membrane and distribution of the lipid and leads to the maintaining of viability of this plasma membrane, and distribution of these protein in the membrane and lipids unevenly leads to the asymmetry to the membrane. So this module is divided into following section. First we will have introduction about the membrane, then we will discuss about membrane composition and asymmetry, then we will discuss about significance of the membrane asymmetry, and we will discuss about hydropathy plot and different hydropathic scales and their significances. First of all, let's have introduction of cell membrane. So cell membrane is a biological membrane or plasma membrane which is selectively permeable to ions and organic molecules. It separates interior of the cell from external environment, and the movement of the substrate in or out of the cell occur through this membrane, and this membrane is consists of phospholipid in the form of bilayer and the protein embedded into it. The membrane serves number of biological and cellular functions such as cell adhesion, ion conductivity and cell signaling. It also serves as an attachment surface for various extracellular structures which include cell wall, glycocalyx and intracellular cytoskeleton. Cell membrane is selectively permeable and able to regulate what enters or what exits from the cell, and hence facilitate the transport material needed for the survival. And the molecule movement across the membrane can be either passive, which doesn't involve energy, or active if it involves energy for the transport. The membrane also important for maintaining the cell potential. Therefore, the cell membrane is important for viability of the organism, and the protein and lipids which is present in the cell membrane are important to study, and their uneven distribution or their asymmetry is also important to understand. The cell membrane, which is selectively permeable, so the role of asymmetry is also important to monitor, to inculcate the selective permeability in the membrane. The cell use a number of transport mechanisms that involve biological membranes. The first one is passive osmosis or diffusion, second is facilitated transport, third is endocytosis, fourth is exocytosis. So the first is passive osmosis and diffusion. Some substrate such as small molecules and ions, namely carbon dioxide, oxygen, can move across the plasma membrane by diffusion, which is a passive transport process. Because the membrane acts as a barrier for certain molecules and ion, they can only passively osmosis or diffuse if there is a different concentration of these molecules across the membrane. Such a concentration gradient across a semi-permeable membrane set up an osmotic flow of the water. The second transport mechanism by the membrane is facilitated transport. The transmembrane protein channel and transporter facilitate the transport of nutrient such as sugar, amino acid. Such molecules diffuses passively through proton channel such as aquaporine, which is used for water transport. In the facilitated diffusion across a transporter or facilitating protein. Proton channel protein such as channel proteins, one example of it permease, are usually quite specific and recognizing and transporting only a limited food group of chemical substrate. Other facilitated proteins such as permeases also specifically transport a limited substrate. They also undergoes facilitated transport. The other mechanism of transport by the membrane include endocytosis, where plasma membrane creates a small deformation inwards, that is known as invagination, in which the substrate to be transported is captured, and this invagellation then pinch off from the membrane on the inside of the cell, create a vesicle containing captured substrate. So endocytosis is a pathway of internalizing the solid particle. It requires energy and it is an active transport. The last mechanism is exocytosis. So exocytosis occur in various cells to remove undigested residue of the substrate brought in the endocytosis. Substrate brought in by endocytosis, or to secrete the substrate such as hormones and enzyme from inside of the cell to outside. It will also transport substrate across the cellular barriers. In the process of exocytosis, the undigested waste containing food vacuoles or secretory vesicle budded from Golgi apparatus is firstly removed by cytoskeleton from interior of the cell to the surface. The vesicle membrane come into the contact with the plasma membrane of the recipient. The vesicle membrane come in contact with the plasma membrane. The lipid molecule of two bilayer rearrange themselves and the two membrane are thus fused. After discussion with the functions of membrane, let's have a discussion of what is a composition of membrane and how the asymmetry of the membrane is influenced by the composition. So the membrane asymmetry represents the difference in both side or leaflet of the plasma membrane, because you know that plasma membrane consists of two lipid bilayers, or you can say two lipid layers. The membrane asymmetry is mainly governed by uneven distribution of lipid or protein in both sides of the plasma membrane. When you are saying asymmetry means both sides are different. So lipid of plasma membrane are distributed asymmetry between two leaflet, and the presence of glycophospholipid and sphingolipid also contribute to this asymmetry. The RBC, or you can say human red blood cells, CARP cells, or you can say our cells, are considered as an ideal candidate for membrane studies. These RBCs are functionally specialized for transporting oxygen. In order to maximize oxygen capacity, it has no nucleus and organelle and consisting primarily the plasma membrane and hemoglobin. Interestingly, when scientists look at the lipid bilayer of RBC, they found that phospholipid composition of individual monolayer are quite different. That means the phospholipid which is present towards the outer leaflet of the plasma membrane is different from the inner leaflet of the plasma membrane. The monolayers exhibiting lateral heterogeneity, where a specific lipid and protein cluster together in a patchwork fashion. This diversity was enhanced by observation of transfer asymmetry of the lipid and protein into leaflet of the bilayer. So you can say that the membrane have lateral heterogeneity, or indirectly you can say asymmetry, and transfer asymmetry. If you see specifically the lipids, you will find that out of all the glycerophospholipid, phosphatidylcholine is located mainly on the outer or exocytosolic leaflet or surface. The phospholipid with terminal primary amino acid group such as phosphatidylserine, phosphatidylethanolamine, as well as minor lipids such as phosphatidyl-inositol and phosphatidic acid are mainly abundant in the inner leaflet. Similarly, sphingolipid, sphingomyelin, and glycosphingolipid are also confined to the outer leaflet. So you can say that some phospholipids or other lipids are located exclusively on outer leaflet and some other are located exclusively on the inner side. Therefore, they will make asymmetry in the membrane. If you see the cholesterol present in the membrane, you will find that cholesterol is distributed evenly throughout the membrane. So you can say that cholesterol is not going to contribute in the asymmetry of the membrane. Although most phospholipids are neutral at physiological pH, phosphatidylserine and phosphatidyl-inositol have a negative charge at physiological pH. And you know that because they are located in inner leaflet, so predominantly these two lipids generate a significant difference in the charge between two leaflet of lipid bilayer because they are negatively charged. So inner side of the plasma membrane is negatively charged. This generates a relevant asymmetry in the membrane where outside is relatively more positive and inside of the membrane is relatively more negative. There are also possibilities of transportation of the lipid from one leaflet to other, and that is known as flip-flop movement, where flip being the movement of extracytosolic leaflet to cytosolic leaflet. When you're saying extracytosolic, that means outer leaflet, and flop being the reverse, that is from cytosolic leaflet to extracytosolic or outer leaflet. Since the polar head of glycophospholipids and sphingolipid make it difficult to transfer through a hydrophobic interior membrane, a spontaneous transbilateral movement is very slow in protein-free model membrane. Therefore, they require specific enzyme that they are known as lipid translocases or flipases for the transport even in the lipid membrane. Recent study has identified 3 protein families with lipid transport locase activity. First subfamily are P-type ATPases, which is also known as aminophospholipid translocases, which flip phosphatidylserine and phosphatidylethanolamine specifically. The second family is ATP binding cascade transporter that flop glycophospholipid with little selectivity. And third family is lipid scramblases, which randomize the transbilayer distribution of all glycophospholipid classes. So in this way we have 3 types of translocases. One flips some phospholipid, other flops some phospholipid, and some other which randomize some phospholipid. In contrast to the asymmetry within the plasma membrane, the glycophospholipid distribution between two leaflet of endoplasmic reticulum is almost symmetrical. Although glycophospholipid are synthesized in cytosolic side of the endoplasmic reticulum membrane, but membrane of endoplasmic reticulum is symmetrical for this particular lipid is due to the rapid translocation of lipid which is independent to the metabolic energy. And this finding leads to the idea that glycophospholipid translocases must exist in the ER, which makes them symmetrical, distributed in the endoplasmic reticulum. It was also seen that liposome is like mimicking structure, containing only transmembrane peptides or protein, so no specific translocase, exhibit a flip-flop of fluorescent label phospholipids, which shows that flip-flop movement of the phospholipid takes place even in absence of enzyme. Moreover, incorporation of the cholesterol into the liposome has an inhibitory effect on peptide flip-flop movement, which shows that cholesterol inhibiting the flip-flop movement. So when you have a membrane, it also have cholesterol, so you need some enzyme for flip-flop movement. Cholesterol is less in endoplasmic reticulum membrane but abundant in the plasma membrane. It again signifies why in the ER membrane most of the lipids are symmetrically distributed. After discussion of membrane asymmetry, reason of membrane asymmetry, let's have discuss what is the significance of this membrane asymmetry. So this membrane lipid asymmetry is important for signal transduction inside the cell. For example, the phosphatidylserine, which is found in the inner leaflet, is a binding partner for signaling protein, and that is protein kinase C. An appearance of phosphatidylserine on the outer leaflet of the cell membrane is an indication of loss of membrane integrity, which are usually seen when there is an apoptosis and other processes which alter or ruptures the membrane. So extracellular expression of this phosphatidylserine target the cell for engulfment by macrophages, because macrophages cleans all ruptured cells, and it is very important, and it is used as a diagnostic marker for apoptosis. Apoptosis means programmed cell death. When cell undergoes death, it follows apoptotic pathway. Maintaining membrane lipid asymmetry is therefore highly important for cell homeostasis. Inositol 4-phosphate. So increased level of this phospholipid in the plasma membrane generally reduces the actin binding and depolymerizes activity of actin depolymerizing factors. The second form of this phospholipid is phosphatidylinositol 4,5-bisphosphate, that is also known as PIP2. So increased level of PIP2 in the plasma membrane inhibit actin filament capping by capping protein and generally reducing the F-actin binding depolymerizing activity of ADF, that is actin depolymerizing factors. The third phospholipids are phosphatidylinositol 3,4,5 triphosphate, that is known as PIP3. Phosphatidyl 3 kinases, that is also known as PI3K, and P10, that is phosphate and tensine homolog signal transduction pathway. In short, PI3K and PTN signal transduction pathway regulates the level of PIP3 in response to extracellular stimulus. The accumulation of PIP3 in philopodia is suggested to cause actin polymerization and increase cellular movement. The membrane asymmetry in lipid and protein composition leads to the proposal that is known as bilayer couple hypothesis. In this hypothesis, it is stated that two monolayer of membrane bilayer may respond differently to various forces while remaining coupled to each other. This hypothesis is the basis for possible shape change observed during the membrane. Maintenance of proper lipid asymmetry is required for mechanical stability of the membrane and for vesicular transport. On the other hand, local or globular change in lipid asymmetry are important for cell cycle progression, apoptosis, and platelet coagulation. Three class of lipid translocases, P-type ATPase, ABC transporters, scramblase are known to be involved in regulation of lipid asymmetry. What is the physiological and pathological function of lipid asymmetry? Lipids of the plasma membrane are distributed asymmetrically between the two leaflets. An activity of three translocases and their regulatory protein make them large contributor of asymmetry. For example, flipping of phosphatidylserine and phosphatidylethanolamine is mediated by P-type ATPase. This is also known as aminophospholipid translocase. The flopase of glycolipids is catalyzed by ABC transporters. The scramblase randomize and collapse the asymmetry of the membrane by randomly transporting some lipid between two layers of the plasma membrane. In yeast, CDC50 family member are also known to regulate component of P-type ATPase. So in this way we can see that these are the proteins which are involved in maintaining the membrane asymmetry, are involved in maintaining the symmetry of the plasma membrane by transporting some phospholipids between the membrane leaflets. After discussion of role of lipids in membrane asymmetry, let's have discussed the role of protein in the membrane asymmetry. Important plot that is known as hydropathy plot, which signifies the role of particular amino acid in protein or in membrane protein. It also explains the stability of a transmembrane helix into the lipid bilayer. So hydropathy is a term that refers to the spectrum of amino acid side chain in hydrophilicity and hydrophobicity scale. It is originally given by Kite and Dolite, which describe an algorithm that identifies helical transmembrane space and that comes to be widely used plot. The hydrophobic scale is based on the transfer free energy from water to vapor of model compound for amino acid side chain, as well as exterior-interior distribution of amino acid in protein structure. These free energy chains can be correlated for unfavorable hydrophilic contribution due to the polar atom and protonation deprotonation event of charged residue upon transfer to a non-polar phase. The alpha helixes can be identified in the protein structure of a membrane protein with relatively high confidence using this hydropathic plot in which each residue assigns a characteristic hydropathic value. Hydrophobicity scales are values that define relative hydrophobicity of amino acid residues. More positive hydrophobic values or more positive hydropathic scale means more hydrophobic amino acid, while less hydropathic value of amino acid indicate it is less hydrophobic or more polar. So these scales are commonly used to predict the transmembrane alpha helixes of the membrane protein. Usually what happens, if you have a membrane protein, the protein is going to embed inside the lipid bilayer, or you can say that if it is a lipid bilayer have a proteins. So if protein is going to have polar surface, as you have in the case of globular protein, that polar surface will not interact with the lipid. So what happen? Usually if a transmembrane protein is embedded into the membrane, it is having intermediate domains which is having exposed hydrophobic groups. But in the case of channel forming protein, the case is more difficult, because channel forming protein are in such a way that they have to stabilize into the lipid bilayer, but they have to also allows molecule to pass through this protein. Molecule is polar in nature, lipid is hydrophobic in nature. So the channel forming membrane protein have a unique distribution of hydrophobic and hydrophilic residues, which makes hydrophobic side chain towards the lipid side of the membrane and hydrophilic side chain towards the core of the channel. And this specific arrangement of hydrophobic residue towards the membrane and polars to the channel is known as amphipathic arrangement of amino acid in channel forming membrane protein. When we are monitoring the amino acid in the protein, a specific region of hydrophobic patches and polar patches will explain about the membrane interacting part of the membrane protein. The hydrophobic effect represents the tendency of water to exclude non-polar molecules. The effect originates from distribution of highly dynamic hydrogen bond between molecule of liquid water. The polar group such as hydroxyl group in methanol do not cause hydrophobic effect. However, a pure hydrocarbon molecule such as hexane cannot accept or donate hydrogen bond to the water. So addition of hexane into the water causes disruption of hydrogen bond network between the water molecule, and the hydrogen bond are partially reconstructed by building a water cage around the hexane molecule, similar to that in clethrate hydrate formed at lower temperature. You can compare this case with that clethrate. So mobility of water molecule in the case, or you can say the solvation layer, is strongly restricted, and this leads to the significant loss of translational rotational entropy of water molecule that make the process unfavorable in terms of free energy of the system. A table comprises 4 different scales of hydrophobicity of amino acid residues in a protein, with most hydrophobic amino acid in the top and most hydrophilic amino acid on the bottom. In this table, you can see that isoleucine is having highest hydropathic index of plus 4.5 and arginine having least hydrophobic index and the value is minus 4.5. So you can see that in the top of this hydrophobic scale we have hydrophobic amino acid. When you move from top to bottom, the hydrophobicity decreases. For example, isoleucine is having highest hydrophobicity, followed by valine, followed by leucine, followed by phenylene, then cysteine, then methionine, then alanine, and the least hydrophobic is arginine. But if you're talking about the hydrophilicity, the reverse is correct. So isoleucine is highest hydropathic index is hydrophobic in nature, while arginine with least hydropathic index is hydrophilic in nature. So other properties associated with this hydrophilicity and hydropathic scale is explained in the table. After discussion of hydropathic scale given by one group of the scientists, let's have discuss other hydropathic scale and their significances. One major scale that is developed by Wimley and Whitehall, and this is known as Wimley-Whitehall residue hydrophobicity scale. The hydrophobicity scale developed by physical methods are based on the measurement of different physical properties such as partial molar heat capacity, transition temperature, and surface tension. Physical methods are easy to use and flexible in terms of solutes. The most popular hydrophobicity scale was developed by measuring the surface tension value for the naturally occurring 20 amino acid in NaCl. The main drawback of surface tension measurement is that the broken hydrogen bond and the neutralized charge group remain at the solution ion interface. The another physical property method involve measuring the solvation free energy. Solvation free energy is estimated as a product of an accessibility of atom to the solvent and atomic solvation parameters. The result indicate the solvation free energy lowered by average of 1 kilocalorie per residue upon folding. The Palliser and Perry has explained about 100 scales and found that they can use them for locating beta strain on surface of the protein. The hydrophobicity scale was also used to predict the preservation of genetic code. Tinquer observed a new order of bases that better reflect the conserved character of genetic code. They believed new ordering of bases was uracil guanine cytosine adenine, so UGCA, better reflects the conserved character of genetic code compared to commonly seen order of UCAG. The Winley-White whole residue hydrophobic scale are significant because of two reasons. The first reason is they include the contribution of peptide bond as well as the side chain providing absolute values. The second thing is they are based on direct experimentally determined values for transfer free energy of polypeptides. Two whole residue hydrophobicity scale has been measured. One, the transfer of unfolding chain from water to bilayer interface, which is also referred as Winley-White interfacial hydrophobicity scale, and one for transfer of unfolding chain into octanol, which is relevant to hydrocarbon core of a bilayer. The Stephen White peptide provide an example of whole residue hydrophobicity scale, showing that the free energy of the transfer, that is delta G, from water to POPC interface and N-octanol. These two scales are then used together to make whole residue hydropathy plots. The hydropathy plot constructed uses delta G difference shows favorable peaks on the absolute scale. This corresponds to known TM helices. Therefore, the whole residue hydropathy plot illustrates why transmembrane segment preferred a transmembrane location rather than surface one. So in this way, the different hydrophobicity scales, and they have very much significance in terms of locating the hydrophobic protein in the membrane as well as locating the distribution of amino acid in transmembrane regions of the membranes. Let's summarize what we have discussed in this module. We have discussed about asymmetry of the membrane. We have discussed asymmetry of membrane arises from uneven distribution of phospholipids and protein in the membrane. We have discussed the transfer asymmetry of the lipid and protein in two leaflet of the bilayer comes from distribution of phosphatidyl in outer leaflet, while phosphatidylserine and phosphatidylethanolamine as well as phosphatidylinositol and phosphatidic acid are in the inner leaflet. Some molecules such as cholesterol are distributed throughout the two monolayers. We have also discussed about the channel forming membranes protein, and we have discussed how the M-pathy present in the alpha helix or in the channel forming proteins are important for the transport of the molecules across the membrane and their stability into the membrane. We have in this module we have also discussed hydropathic plots and their significances in determining the future of a particular peptide in the membrane. We have also discussed the different hydrophobicity scales that are also into use. So in this module we have discussed how asymmetry is important for the membrane, and we have also discussed how hydrophobic plots are used to predict the future of a peptide or protein or membrane existence.